Hello, everyone. We'll give it just another minute here.
Hey, good morning, everyone. Thank you for joining. We're going to give folks just a few moments to dial in. As we're getting settled, we're going to put up a poll. If you wouldn't mind taking just one moment, putting an answer to that, we'll get started here just momentarily. All right. Well, I think we've got a pretty good group here so far, and I apologize for not being on video here. Somewhat of a Zoom technical difficulty here, so you'll have to put up with my static face. Again, to kick things off, welcome everyone. Thank you so much for joining today. We're excited to be here with all of you talking about translating data analysis and research into publication quality discoveries. I know a topic near and dear, or at least familiar to many of us. My name is Travis Doherty. I lead our customer experience team here at Pluto. I have the distinct pleasure of working with scientists, labs, and departments from all over and supporting their research endeavors. And I'm seeing some familiar names in the attendee list here, which is awesome. I'll be moderating today's webinar, as well as the Q&A session that we'll do towards the end. This is really meant to be as informative and as valuable to you all as possible. So you all should notice in the webinar here a Q&A button. Please, as we go, submit any questions that come up throughout the presentation as you think of them. Again, we'll dedicate the back half of this towards that Q&A session. So it is now my pleasure to hand it over to Pluto's founder and CEO, Ronnie Powers.
Hi, everybody. Thank you so much for taking the time out of your day to join us today. I think we have some really exciting content, great tips and takeaways for everybody. For a little bit of background context on myself, I started my career in the wet lab as a molecular biologist. Have really just always been fascinated by molecular biology data, some of which we'll be talking about today. But really recognizing this need based on large data sets for better computation and better tools to enable biologists like myself, I earned a PhD in computational biology and have really since then made it my mission to make omics data analysis and understanding and interpreting molecular biology data a lot more accessible to the hundreds of thousands of biologists around the world. And so it's really a pleasure that we have at Pluto to be able to work with some top tier, really talented scientists. I'd like to share during this webinar today the tips and tricks really that we have learned a lot over the past few years about data visualization, finding and analyzing omics data sets, creating figures that we all know will lead to higher impact publications, as well as better grant funding, which I'm sure is top of mind for a lot of you all in the audience today too. So I think we have a really great agenda planned. In terms of this, mission that we've talked about, the mission really to inspire more biologists like yourselves to use multi-omics data sets and really make these impactful discoveries is one that's very personal to me and the whole Pluto team. We would love to take a really quick moment and just throw up one additional poll here to understand, really what's important to you in your research. This will help us adjust just slightly, make sure that we're really tacking on here tips that are going to be most useful to everybody. So take a quick second, drop an answer in this poll. You are able to select multiple answers, so feel free to choose, the one or many that are applicable to your own research. We'll really get started here, diving into our agenda. Excellent. I see a lot of great answers coming in. Looks like we're hitting a lot of these topics and categories. Our top winner at the moment is the ability to do my own analysis. So this is really great, and once again, near and dear to my own heart and Pluto's mission. We'll be talking through analyzing multi-omics data today during the webinar. We'll be showing you how you can do that in our platform, Pluto. But just a note that these takeaways and tips will really be applicable regardless of the process that you currently use. We try to just share these learnings and make ourselves available to you all and to the scientific community as a resource. So I'll go ahead and close down this poll. 2 more seconds. Perfect. Alrighty. So moving forward then into our agenda for today. So the topic of today is really omics data sets. We'll use transcriptomics as an example here. Epigenomics or epigenetics can follow a very similar process. We'll kind of start our journey talking through the process of analyzing data that comes from the GEO database. This would be a common use case, a common way to identify public data sets. We'll also talk about running NGS pipelines, whether that's on published data or your own. So if you've generated those large raw FASTQ files, what does that process look like for running your NGS pipeline? We'll touch briefly on quality control, which is a really critical aspect prior to figure generation that we always want to encourage researchers to take into account. And then we'll talk through performing standard kinds of analyses, tips for creating figures that are really kind of one level up, or this high impact kind of custom feel to them that we think really impacts your ability to win grants and publish higher impact papers. And then a lot about the preparation to publication process. That's a bit of a tongue twister. So thinking about our methods, thinking about how we export high resolution figures, all of that. And just to restate, like Travis mentioned here, please feel free to submit questions in the Zoom Q&A box. So look for the Q&A section. You can submit those questions at any time throughout the presentation. Travis will be fielding those for us during the presentation, and then we'll open it up for Q&A probably in the last 20-15 minutes of this call. All right, so diving right in, you should still be able to see my screen, and we're looking at a publication. All right, so diving into this kind of journey that I mentioned, it's really common for researchers to be, looking through a paper and finding that maybe this data set is relevant to your own research. So I'm looking right now at a paper from a lab, the DARIP lab, which is now at the University of South Florida. And this paper examines some RNA sequencing data, some transcriptomics or gene expression, lots of different words to refer to this, right? Where they were using RNA-seq to investigate cerebral ischemia. Now, reading this paper, you might be familiar with the fact that you come across a GSE ID or a reference to this data set that's been published in GEO. And so I wanted to send over on this tab, just to give a quick view for what this typically looks like. This GSE ID is this GSE followed by several numbers, and it corresponds with that PubMed ID paper that we were just looking at. So this is the data set, one of 2, I think, that are published in this paper. And we do have some brief description of the methods here. So once again, this is a pretty common process where you might see, hey, this paper is relevant to me. I'd like to take a look at this data. And the reason that we use published data when trying to leverage them for our own analysis is often because we're interested in asking different questions than the original paper would have been addressing. So if that were the case for this data set, I might come down here and take a look at the 12 samples that are available in this data set, and then determine, yes, they have these 3 time points, 6 hours, 12 hours, 24 hours. I'm interested also in investigating the gene expression profiles in these different mouse samples, maybe for my own particular research question. Or if this was your experiment in the first place, you might have these large FASTQ files associated with your 12 samples in this experiment. Now, it's completely possible to come into GEO here and find these sample annotations, pull this data, and then even download the FASTQ files through their SRA run selector. Anybody that has tried this process in the past knows that it can be a pretty tedious, not to mention just really large data files take a lot of space on your computer or hard drives or servers. So overall, a relatively tedious process. This is one of the first things that we've really tried to streamline in Pluto. So I'll show you in Pluto, for example, if we had been looking for a particular topic or disease in our software platform, and I'm landing here on my home page, it's possible to just dive straight in here to publish data sets. And here we could search for some keywords. And in this case, pull up 7 different data sets, maybe related to our disease of interest. Now you'll notice that these all have the GSE IDs still on the titles of the experiments. So they let us know that we can always go back, click into them, look at the original geo reference. But the key thing here is that we're able to, with one click, copy this data set and pull it into our Pluto lab space to begin our analysis. So this is really starting with a published data set, and we'll go through the end-to-end analysis process here. But I'll take a quick detour to show you what it looks like if you're analyzing your own experimental data versus a data set from Geo. You'd create new experiment. All right, and then select a project that experiment would go into. In Pluto, projects function very much like folders. And so we'd give our experiment an informative name. So maybe we'll just give it a name like this. And maybe this is mouse data. And then you'll select from an available pipeline. These are all configurable in Pluto on a per workspace basis. So we're seeing the pipelines that I have enabled and the organisms that I have enabled in my current project here, or in my workspace. Now, the 2 components in Pluto to creating an experiment, very much like creating an experiment in Geo, are the assay data and then the sample annotations. So the assay data for, in this case, an RNA-seq experiment that we're using as an example, can take the form of FASTQ files or processed CSV, such as raw counts or other units here. And either of those can be directly uploaded to Pluto so that you can run your pipeline from the FASTQ files or just begin analysis straight away with the CSV files. But relevant to the group of experiments we're talking about today, you can also pull in experiments directly from Geo. So let's say you had searched by the GSE ID for this particular study we were just looking at, and it hadn't previously been imported into Pluto. We have over 15,000 experiments in the platform already, but new ones are getting added all the time. And so all you need to do here is submit an import request, input the GSE ID for your experiment, and the FASTQ files will be deposited from NCBI into Pluto's servers within 24 hours. So this would be the process for creating your own experiment in Pluto, really starting from scratch here, uploading that assay data. And then likewise, the sample annotations are just a CSV file, a tabular representation of the different biological variables in your experiment. And so if I go back to Geo, just to compare, you'll see the format here. If I click into one of these samples, I can see some characteristics that can be parsed out from Geo's website here, such as strain, tissue in this case, and then time point being the main variable that was being compared. So in Pluto, that tabular representation, I think, is a little more friendly. So if we go in to view all of our experiments and go into this experiment that we're looking at, here's the tabular representation of the sample annotations. So this is the same experiment we're looking at in Geo. You can see we have 3 different time points, 6 hours, 12 hours, and 24 hours. And then we have FASTQ files, those raw sequencing files that are associated with each of the samples. So in terms of the agenda that we wanted to cover today, we wanted to talk about pulling in this published data from Geo, which you can do yourself or via a platform like Pluto, associate those FASTQ files to a sample. And now the next step is really converting these FASTQ files into numerical data that can then be analyzed. The FASTQ files themselves contain literally A's, T's, C's, and G's, which aren't able to be plotted just as is. And so, as you're probably familiar, we're trying to get to these raw counts or assay data. So in Pluto, when you upload your FASTQ files, you can kick off an RNA-seq pipeline or one of the other pipelines from a previous screen that I was showing that generates this count matrix that we're looking at here. And you can explore these for individual genes, but of course this is raw count data, so we're really not ready to quite do anything with it yet. But that was a transformed view of these original FASTQ files that were uploaded. So after uploading your FASTQ files and then seeing that this count matrix has been created, it's important to go through and look at the quality. And I will put a plugin here for rerunning public data from FASTQs, even if the authors make published data, the processed data rather, available. We've identified, unfortunately, many datasets on Geo that have poor quality samples. And this is a really important thing that you need to know about before beginning your analysis. So quality of FASTQ files is typically shown in this multi-QC format. You can see a variety of different statistics on, for example, the mapping and alignment. This one's pretty decent. We want to see a large chunk of blue here, showing that a lot of the reads have aligned uniquely to a gene. So the quality control process here is kind of, an entire section or methods in and of itself. This is something that Pluto helps scientists with frequently in terms of understanding what went on in their pipeline here, pointing out, any quality issues. But it's definitely something that I would recommend for everybody before beginning your data analysis. If you're working with a bioinformatics core and they don't generate these kinds of reports for you, you should absolutely request them. In Pluto here, we can also see the methods for the pipelines. Methods are a really important component for publication. We all know that a method section will be in your paper. It's really critical to keep track of these methods as you're going along, rather than at the very end, trying to trace back, for everything that was done. So a platform like Pluto is really helpful in that the methods are dynamically generated and always available. Everything's traceable end to end so that you don't have to worry when it does come time to publish about finding those. If you're using other systems or outsourced, bioinformatics cores or consultants, really make sure that you get those methods up front. The methods in Pluto here include references as well for any open source pipelines that were used, and so that can be one-click copied into your papers. All right, so that was kind of the initial process for, finding data sets potentially from a paper that you're reading or from Geo, pulling them into ideally a system that's going to let you yourself do the exploration. So at this point, we'll move into the more fun part, I think, which is the downstream analysis and how do we actually create these figures and make really compelling visualizations for our publication. So in Pluto, we can do that here. On two different views, we have kind of a dashboard view that we call analysis, but I like our flexible view called a canvas. So here we have now an infinite canvas where initially we are just going to be telling a scientific story. If you're familiar with other platforms that analyze omics data, you might be familiar with more of a dashboard view where you put in raw sequencing data, run a pipeline, and then you're shown a relatively static page that maybe gives you a volcano plot and pathway analysis. And the problem is that many experiments don't follow that kind of pattern. And in fact, in this experiment, we saw that there are 3 different time points. And so I would really like to, as a scientist, be able to compare those 3 time points, overlap them, do a deeper exploration of pathway analysis and top genes, and tell a scientific story that's going to unfold as I'm doing the data analysis. So our Canvas medium here provides a lot more flexible way to do that. This is also something that we see scientists, do in slides or maybe on paper, kind of napkin, draw it out. But I think really encouraging you to tell that scientific story is a great place to start. So here on my Canvas, I'm first going to create a new analysis. A very common first step when looking at omics data would be to just ask, how do all of our samples in our experiment cluster or relate to each other in a low dimensional space? And for that, I can use an analysis like principal components or other dimensionality reduction algorithms. So here are 3 pretty common dimensionality reduction algorithms that will look at gene expression across all 30,000 plus genes, and then map our samples into a low dimensional or 2-dimensional space here. There are a lot of great resources on all of these algorithms. In Pluto, we try to have these blurbs and example plots to let users know and scientists know kind of what to expect. So here, starting with a principal components analysis, you'll see that we have a very limited set of parameters really that need to be adjusted. You can get pretty far, with a lot of these algorithms just using default parameters. So here I have a couple of data transformations that I can choose from, and I'll just leave those as recommended and then click run. This kicks off a tiny bioinformatics script. They all run an auto-scaling infrastructure in Pluto, so you can be doing multiple things at once. Multiple people can be interacting with the same data. But you'll see that it returns back a principal components plot. And so now if I exit out of here, you can see I have this graph available on my canvas. I can resize that, move it around. We're going to tell this scientific story. So the interpretation of this graph, to me, seems pretty clear. We can see that our samples, which I can reorder legend-wise here, cluster pretty well with the expected groups. We have this control group down here clustering very tightly together, and it looks like our 6-hour group also clusters pretty nicely. We see a lot more variance in the 12-hour and 24-hour time points here. And so it might be worth looking into, the biological conditions under which these samples were collected. But this is really just great to know as we're starting our experiment. I like to change, for example, my controls to be at the front and have those be maybe a different color, like a gray. And so these colors can all be edited in Pluto to make those changes a little bit more obvious. All right, so there's our first graph on our canvas. I'll add a couple more and then we'll point out some methods and export. So because we have these 3 different comparisons now, our 6-hour, 12, and 24, or there's our 24 here, we'll want to do some differential expression analyses. So I'll again create a new analysis here, which will be this differential expression type, and create. Back to this aspect of scientific storytelling, you may have likely some different groups within your experiment that that you'd like to compare. So initially, when we're doing something like differential expression, we're just tackling those scientific questions that we had really designed the experiment around. So in this case, I'm going to group by my time point variable here and compare my 6-hour time point to my controls, which were these 3 samples that have an NA value in the time point column. And what you'll notice here in this analysis is that Pluto has pulled out the biological variables from our sample annotations and made those available to me in the user interface. And this is what is allowing me to do this kind of comparison. So I'll click run and run a quick differential expression analysis here. And that produces a volcano plot by default. I can also visualize this data as a heat map. So we have our PCA plot there. I'll quickly run a few of these differential expression analyses for the 2 other time points that we have. And then we'll get back to talking about our scientific storytelling. So I'll select the 12-hour group now, and then do this comparison. Back out to our canvas, kind of move these around, position them the way that we like. And lastly, do our 24-hour group. Now I've talked a bit about the interpretation of these graphs. I'll give a quick note here that Pluto does use an AI, which is this little light bulb icon to the right. And we also have PhD bioinformaticists on staff that are available to help you if you have questions about interpreting your analysis, maybe if you have less experience with these kinds of analyses that I'm showing. Okay, great. And oops, lost my Zoom. Got too many things going on. All right. So now I've created these 3 differential expression analyses. We'll let that canvas load. We'll delete this line that I accidentally created. Okay, great. So now back to this aspect of scientific storytelling. We have these initial figures in place, and I'm going to remove all of these group genes that I accidentally highlighted here. So we have these figures in place, so we can now start customizing them, which I showed you can do with the PCA plot by changing colors. With something like a volcano plot, you can also change colors and lasso top genes of interest. You saw on that volcano plot option that I have a couple of other kinds of appearance customizations that I can do. Let's go into our 6-hour time point here and maybe show some examples. So here I can change things like p-value thresholds. This really contributes to your scientific storytelling process. If you're wanting to indicate, how significant things are, you might want to show a line on that graph. You can see that we have many, many genes that are significant in this particular comparison. And we can customize colors. We can also do things to further our scientific story by asking, what were the top upregulated genes in this particular comparison? So it looks like most of our genes in this case were upregulated significantly, but I can take a look, for example, at what these top 4 genes are, and I can do that by lassoing. It's really common to have a custom gene list or a couple of genes that you're interested in exploring. And these custom gene lists can be saved in Pluto or saved in any system that you're using. And that way you can use this for downstream analysis. So this happened to be 4 that I highlighted here. I could copy this list of genes now down in the left and then exit out of this window. And so another, follow-up step that would be pretty common here would be to plot those 4 genes to understand the variance that we're seeing between samples. So a tip here is thinking about, what a reviewer would really be curious about after looking, at the figure that you've created. So we'll just create a few more figures here to kind of illustrate this process, and then we'll start getting into the refinement and methods and export process. So I'm going to plot normalized values for the 4 genes that I just looked at, just to give another sense for how you could ask a follow-up question here based on your initial analysis. I'll plot this for all time points, actually. This will load up a bar plot. We can customize this graph into different kinds of visualizations. Once again, edit colors, move our groups around here so that I could call this my control group and say, just rename this to control instead of an A, reorder my time points here. Great, and I think I had made this one a bright pink and save. So we can move this along on our canvas here and really, like I've said several times here, tell this scientific story by connecting some ideas as you're exploring your data. So now we've taken a look at this 6-hour time point. Another downstream analysis that would be very common would be pathway analysis. We've looked at some of these top genes to see how variable they are between samples within our different time points that we're exploring. Just to wrap up this exploratory phase that we have here, I'll end by overlapping these 3 comparisons to identify consistent genes across conditions. This is also a great kind of figure that we see published very frequently in papers, where you're asked to demonstrate what's similar and different between different conditions. So I'll create a new analysis and do an overlap here and create. In this case, I'm going to be pulling not from a manual list, which I've mentioned you can save in Pluto, but I'll be using our differential targets list. So I can use 6-hour as one of my comparisons. You'll see the comparisons that I've created here and save. I can use another differential targets list for 12 hours and select that. You'll see that I can edit p-value thresholds and directionality. Also, I'm just leaving these for right now. We'll probably have a lot of significant genes and they're in the up direction. But I'll save that. And finally, we'll add one for our 24-hour time point. So through this process, what I'm really hoping to convey is that based on your data, you should be asking these iterative and kind of step-by-step questions about your data based on findings from your previous plots. And so if you're working with a bioinformaticist or another platform, this process can look pretty static, and it's helpful to continue to ask these follow-up questions and say, based on what I'm seeing in this graph, what is the next thing that I'd like to pursue? We can edit this Venn diagram. I just turned it on so that I can see the names of the groups here, so that we can see in this middle section, we have 1,094 genes that are present in upregulated significantly in the 6-hour, 12-hour, and 24-hour time points here. And like in the case of all of these other graphs, we can edit the colors in Pluto. Now, I know color editing might seem like something trivial, but if you take a look at high impact papers, such as those published in Nature and Cell, you will notice that a lot of thought is put into the color palettes that are being used in figures. So I would encourage you to either explore publicly available color palettes. There are libraries such as Color Brewer or others available to help with contrast, to help with color blindness and legibility. And so that's definitely worth exploring. Unfortunately, some of the open source packages, say for doing differential expression or others, other kinds of plotting, ggplot and things, the default color palettes are really not ideal from a contrast and publication perspective. So take that little extra time, really customize the colors on your figures here. Okay, so now we've drawn out this kind of thought process. I really like this Canvas medium for telling your scientific story. However it is that you're explaining this medium to yourself, to your trainees, really think through that scientific story that you're wanting to tell. And now in Pluto, when you've decided on ordering for figures, you can combine those figures here on that canvas also, or leave little notes and emojis for yourself about maybe which graphs should be included. So at this point, I'm going to shift into, saying, okay, we've gotten this story down pat. We're really excited about creating figures from these data sets. How do we kind of wrap up and think about final steps for taking this thought process that we have and turning it into our final figure? So a couple of things that you'll need for publication. First of all, every analysis in Pluto has tabular results associated with it. Every analysis that you're doing, I should say, regardless of platform, will have tabular results. And so for a differential expression analysis on the results here, you'll probably be familiar with a differentially expressed gene table, which you can explore. Some publications will require you to download this table and provide it as a supplemental file. So remember to do that. Method sections also we've already touched on a little bit, but I'll just show that for each of the analyses that you're doing, it's also important to include these method sections. You'll see that the ones in Pluto are dynamically generated, so they're relevant to this particular comparison that we were looking at, and they include any threshold values that we had set during the analysis. So make sure to keep track, of the parameters that you're using, even if you're exploring analyses, maybe changing and tweaking things over time. For whatever this final set of figures is, make sure that you have these in mind. And then if you're using any kinds of R packages or scripts or underlying code to do these transformations, because they all depend on code, everything that we've shown really can't be done in Excel, except maybe the bar plot. So make sure to retain references for those underlying R packages, such as DESeq2 in this case. Great, so that's the tabular results section. Another critical aspect will be and we talked about the methods also, kind of our last critical aspect here will be actually exporting the plot for publication. So I'll move over here and just show our classic analysis view. Since I mentioned it before, this is just a more static or more kind of grid-like view. I can still move these graphs around. But here from this view, I'll show that we can export these figures. Let's see, I think the PCA will be, I'll use this one maybe as an example. So if we're taking a look at this bar plot, you'll see that we have this ability to export the graph. Now, exporting graphs is not so simple as just, taking a screenshot of the graph that you're seeing on your screen. We know that depending on how you're going to use that graph, you'll need to have a different layout. So if you're presenting this bar plot on a slide, you may want a very long format, depending on the other content that you have on the slide. Similarly, if it's part of a figure, it may also be worth, showing it as this skinny kind of panel here so that maybe you have some additional figures nearby. And then your legend may need to be in different locations also. So you'll see I have edit control over those aspects here in Pluto. This is another common layout that I see. I think really the takeaway here is always ensuring that your legend or this key rather, is visible, that you have those high contrast and colorblind friendly colors that we were talking about. And then really making sure that it's easy to connect the two, the legend here, as well as the data that's being shown. You can edit axis labels and colors as well, and make sure that all of these are ready to go for your publication. I didn't edit the title on this graph, but it probably would have made sense here to say, top 4 most upregulated genes, and that's something that I could have done on the prior screen. But once your figure is in a good place, our last tip here is really to download that figure in a high resolution format. And so in Pluto, for example, you can download it as a high-res PNG. An SVG is a vector format that you can take directly into Illustrator and then use this figure alongside any other panels that you're compiling. The SVG vector format does allow you to continue editing things like text and colors. And so that, is definitely useful for figure preparation. We also offer this PowerPoint and DocX format for export. These ones are unique in that they come with the methods section that's associated with the graph. So you'll have a separate text box on the slide, on the PowerPoint slide. If we're downloading in this format, you can obviously choose to delete that method section, but it's nice to have it right there alongside your graph so that you can manipulate or use it as a caption. And then same thing with the DocX format here. So let me take a quick look at my notes. I think we've talked through all of our, polishing figures, this process of first telling a compelling scientific story based on the question that we had in our experiment, looking at the tabular results that are produced because we'll need to turn those over for publication, customizing figures to really ensure that they have the look and feel that we're trying to achieve, and really draw attention to the key results. And again, that key story that we're trying to tell. We've talked about method sections, and then one final aspect of large data sets, especially in these NGS data sets that I wanted to point out, is making sure that you have a plan in place for the NIH data management and sharing standards. So we started this presentation by showing a paper and by showing data that had been deposited in GEO. And likewise, when you are analyzing your own experiment, this will be a process that you'll have to follow to return your data to the public domain here in GEO yourself when you're ready to publish. And this is a process that can be pretty tedious for bioinformatics cores or for an individual researcher to complete. And so I did want to point out here that in Pluto, on our workflow tab, you can see our R package and Python package for interacting with the platform. But down here in the other connectors section, you can start a GEO submission for this experiment. So because all of the raw data, the sample annotations, everything is already here in Pluto, you can click this button to have a submission drafted, which you'll of course be able to provide final approval for, select an embargo date, and some other things before Pluto initiates any transfer to GEO, but the process saves a substantial amount of time. And then you'll be able to see your data shown here on GEO when your publication goes live. And that way it's available for others to analyze and leverage for their scientific questions as well. So I think we're doing really well on time. I'll just jump back over to our slides here. And we can open it up for Q&A. So let's just check. We have kind of run through all of these aspects. Great. And so major takeaways and tips really for the high impact analysis and visualization that we covered in this short 40 minutes here. We really walked through leveraging your own experimental data, identifying relevant published data sets on GEO, pulling in those fast queues to run your own pipeline, and the importance of QC when you're looking especially at sequencing data, telling a compelling step-by-step scientific story, and making sure that you're following the data, that you aren't just looking, at a static dashboard or looking at graphs that maybe somebody has told you, are related to RNA-seq. Really doing one graph, thinking about it, and then doing the next graph. Once you have those graphs, customizing figure appearance, making sure that you export them in a high resolution for publication, and then collecting that tabular data, supplemental materials, as well as the methods, ideally as you're going along and having that all be traced. And we ended with showing you really, a reminder to keep in mind these NIH data management and sharing guidelines. So think through how you will be submitting your data to GEO during or after publication. And kind of related to that, if you have used any published data from GEO, make sure that you're able to cite that in your own paper and cite any other tools and references used. So I'll go ahead and stop there and turn it over to Travis now for some Q&A.
All right, great. Thanks, Ronnie. Yeah, a number of good questions already in the chat. Anyone that has other questions, go ahead, type those into the Q&A button at the bottom, and we'll start to run through those. So one of the first ones here, and this was submitted early on when we were looking at a GEO study. What do I do if FASTQ files are not available in GEO for a study that I want to investigate?
That's a great question. So in many cases, FASTQ files will be available on GEO. One important thing to note is that, if I can get out of my slides here, is that you may not see the FASTQ files available here. They won't be listed here. You have to kind of go through this SRA run selector process typically to see these files. And so if I click this and then go into, this one may actually be an example of what we're talking about. You'll see that the FASTQ files aren't showing up here. So NCBI Geo has this deliver data functionality that you can use to get the FASTQ files directly from their cloud buckets into your own cloud buckets. And so first note on that is just, if you don't see the FASTQ files available on Geo, it doesn't necessarily mean that they aren't there. However, if you do go through the process of requesting the data delivery through a cloud bucket on Geo, and that process tells you that the FASTQ files aren't available, you have a couple of other options. You can, of course, go directly to the authors. We reach out sometimes on behalf of our clients to original authors to request that the data be made available in some kind of sharing mechanism that works for both parties. There may also be alternative file formats like a BAM file that you can use to either get back to a FASTQ format or just carry on for analysis from there. That's also a little more ideal than processed data because you can get at least some quality information from that. But as kind of our, like a final resort, which isn't a horrible one, but a final resort would be to use the processed data that they've made available. And so that might look like, let's see, in here, this one has GTF files available, but sometimes you'll see that there's a text file or a CSV file on Geo. The thing about using processed data, just make sure that you confirm the units. So it's very important to know if it were raw counts that were used, or if they were TPM values or FPKM values. Those all have different statistical assumptions that will determine downstream analysis. And so make sure to make a note of that and use those statistical assumptions as you're selecting different analyses. In Pluto, if you select a unit such as TPM, you'll see that that analysis catalog that I was showing downstream actually changes slightly depending on the units that you use. So those are my recommendations. We've definitely run into all different kinds of experiments of shapes and sizes that when we've been working with Geo.
Oh, great, thanks. One question that came in, Ronnie, I believe you were showing one of the volcano plots earlier. How do I find a list of all these genes and export it to an Excel sheet?
Sure. So from the volcano plots, or I'll go back to our classic view here. So from the volcano plots, that tabular data that I was talking about, a DEG table, contains all of the genes ranked by their adjusted p-value. So on this 24-hour time point, for example, if I view the results, this is where you'll see for every gene that was measured in the experiment that was also assigned a p-value by the DESeq2 library, in this case, 20,000 genes. These can all be downloaded and searched. If you're running the DESeq2 R script yourself, same thing, you would get a CSV file that looks like this. These are ranked by adjusted p-value, and so the most significant ones are shown at the top here, but you can see we can go back many, many pages and find even non-significantly up or down regulated genes. Here's the overall count up here.
All right, great. Another question that just came in, is this good for metabolomics data?
Mhm. That's a great question. So metabolomics data follows pretty similar statistical assumptions as, say, a microarray. It's a little bit different than sequencing-based data, but you are able to analyze it in Pluto or similar platforms following a really similar process that we showed. Your raw data files won't be FASTQ files. In Pluto, you'll start with a CSV file of abundance values for metabolomics. You upload that CSV file along with your sample annotations. These can follow any format. So in this example, we had, some different time points that we were looking at, but you would just put any kind of experimental annotations into any columns that you needed, and then upload a processed CSV file that looks a lot like this raw count matrix, where you have your metabolites annotated typically with a KEG ID or other identifier in the first column. And then these subsequent columns are your samples and their abundance values. Downstream analysis looks very similar. You can perform principal components and other dimensionality reduction. When you're doing differential abundance, it does use a different library. So in Pluto, we use Lemma for analysis of metabolomics and microarray data because of the statistical assumptions in that data set. And so you can just see that reflected in the methods. So you would pick differential abundance from the catalog. You would do this very similar kind of analysis. But then if you open up the methods, you would see those different statistical assumptions and the R package used.
Wonderful. Here's one that we run into often, just given the nature of what we do. Is there a max size for FASTQ files? I have close to 400 gigabytes for my experiment. Can they be uploaded seamlessly?
Mm-hmm. Yep, definitely. This is a challenge that, like Travis alluded to, we run into quite a bit. So Pluto integrates with university servers, with all kinds of storage systems, cloud-based, GCS buckets and AWS buckets. So the first challenge really is, getting the data into the system. So yes, we've tried to make it as easy as possible to get those large FASTQ files transferred into Pluto. FTP is another mechanism. So we deal with that part all the time. In terms of the overall limit, our platform as a cloud-based platform is based on an annual subscription. And so your file size limit is essentially just built into your platform tier. So that would just depend on, the tier that you're on, and then how many experiments you're running, how many samples you're running per year. But we frequently deal with really large sample sizes and really large FASTQ files. The single cell data sets that you can analyze in Pluto, typically a FASTQ file will be 8 to 30 gigs just for a single FASTQ file. And because we're based on the cloud, we have theoretically infinite scaling. So there really aren't any limits on our side as far as the size of data that we can support because we use this auto scaling infrastructure.
Great, thank you, Ronnie. A question that came up, this is as you were outlining the QC report in the experiment that we were showing. What do I do with samples that have failed the QC?
That's another great question. So that will really depend on the goal of the experiment that you're working with. So if the experiment is really intended to go into publication and you've identified a sample that doesn't meet those quality criteria, ideally you will have other replicates that you can use for that comparison that you're hoping to do, in which case you can just remove that one sample. Now we know that sequencing experiments are expensive, and so you really want to avoid completely throwing out a sample whenever possible. So there are, thresholds really to that quality. If you're seeing a sample that just has lower sequencing depth than the other samples, something like that can be accounted for with a counts per million normalization. So you can use some normalization strategies to adjust. Likewise, if you're finding a strong batch effect in your data, if you're seeing that samples that were sequenced at particular times are exhibiting differences that aren't due to a biological condition difference, you can also do a batch normalization to help make that adjustment before then doing downstream analysis. It really is just kind of, a nuance and being transparent, with the methods of your paper about what thresholds were used, the rationale for selecting different samples or removing some from the analysis. And that's, again, something that our bioinformaticists deal with quite a bit. And so we can share, best practices and recommendations around that.
Awesome. Thanks, Ronnie. Here's one that I can go ahead and answer, and I imagine others were wondering as well. Is the seminar recorded? Can we have access to the recording? Yes, absolutely. this will be shared out with the group as soon as we close out today, as well as with some other resources. So we'll make sure that you get that. Let's see, moving up here. How can we do gene ontology analysis in this platform?
Sure. So I'll go back into our experiment here, and this should be pretty quick to show. So maybe just from our classic analysis view here, you can use a Go-term gene set collection when doing GSEA pathway analysis. That's frequently how people would do the kind of analysis that I think you're referring to here. So if I add an analysis to my page, I can filter down by our pathway analysis category. You'll see we have different kinds of pathway analyses. You can focus in on just transcription factors, but this gene set enrichment analysis relies on collections of gene sets that have been previously annotated by different entities. And so Go, which is actually shown in this example here, or gene ontology, is one of those such collections. You can see here different kinds of graphs that we'll be able to create with this analysis. And so if I create this analysis, I would select a comparison, like our 6-hour comparison, and then select the collection. And this is where you can find here C5s. These are all the gene ontology gene sets. And so I always get it mixed up if it's biological process or molecular function. I want to say biological process is maybe a little bit more useful. So you would select that gene set. You can edit some advanced settings here if you'd like in the GSEA, but then you would just click run. This analysis I know takes probably 30 seconds or so to complete. It's going through a list of, I think, a couple thousand gene sets from the gene ontology, and then it will produce results for us that way.
Which actually segues nicely, Ronnie, is there's an unrelated question. How many analyses can I run at one time?
That is a great question. So any number of analyses. So you see that one is currently running. This, I can kind of move around on the page. It's an independent entity, but I can continue adding analyses. So I could add, any of these other types of analyses that we have to pull from. You can also add static content like images, Prism files, which can actually be previewed. And so if you're doing some analysis in Prism, you can pull those in, spreadsheets, do correlation analysis. So any of these, we can click into and keep doing that analysis and just have multiple of these little widgets that are all running on the page. I'll take this opportunity though to point out the kind of Google Doc style sharing that you have in Pluto too. So I had created this experiment in a team project. And so you can see my team here automatically has access to my experiment, but I can add and invite collaborators here with a view or edit role to just this page. And any of the editors that are shown here can also at the same time be here on this page, making their own edits. And so they could be, Andrew could come in and Travis could come in and we could all be running different analyses. We can comment on each other's analyses. So you can leave comments in this little comment bar here to have conversation threads related to your different analyses. And importantly, so that this doesn't just sound like chaos, on our collaboration tab here, you can see all of that change history. So it's very clear to everyone that has access, who has the ability to edit, and then you can see that I've been making a lot of changes here on this graph, but if somebody else was involved, you'd be able to see that too.
Excellent. And there was, it looks like actually a clarification on that last question, Ronnie. How many analyses can I run in an experiment? I think that's more with regards to the analysis limit.
Oh, sure. So there's really no limit on analyses. On this page that we're looking at here, you can add 20 at a time, and then it will just have you go to another page. But you can keep going infinitely on this page. And then likewise on our canvas view, this is an infinite canvas. So you can see my little mini map up here, but I can zoom out and I could really lay out, this whole analysis that I'm doing. I can move over here and do a completely new analysis. So it's, we frequently will see users have, 60, 100 plots that they're exploring on their page just because there's a lot of data with 30,000, 20,000 genes to look at.
Excellent. And thank you for clarifying that question. I think we have
Mhm.
time for a couple more here. This question was submitted, Ronnie. We were looking at the experiment you stepped us through, specifically on kind of the assay data and the sample data. The question is how
Mhm.
to remove one replicate that did not work.
Mhm. Yep, so that's a great question. If you get to your, pipeline step that we had talked about, and then decide that one of those samples isn't going to move forward in analysis, you can edit your sample data, and just in this table, you would remove that row from your data. And then your downstream analysis won't take that one into account, but the original FASTQ files and QC and everything will be retained for that sample.
Awesome.
That would
Um,
be how to do it really at the global level. You may have noticed also that on some of these individual analyses, I could select down to just specific groups of samples. So if I wanted to remove that sample just from my PCA plot, I could do that also.
Great. Thanks, Ronnie.
Great.
And I think time for one last question. You stepped through different analyses during the exploratory process you demonstrated. Does Pluto offer guidance on analysis to run based off the data we are investigating?
Yes, absolutely. This is so core and central to our mission and something that I'm extremely passionate about. So absolutely, you can use our AI tools to do that or talk with our bioinformaticists. So an example workflow there might be, looking at, say, doing your first differential expression comparison, and then chatting with the built-in Pluto AI to ask, what does it mean that I have so many genes upregulated, or are there any patterns in these genes? What would be a good next step? And our AI will suggest probably, a pathway analysis to understand pathways contained in these upregulated genes, or plotting these. So we're really training this system to act a lot like a scientific co-pilot, exactly the way that I would go about, analyzing an experiment or helping somebody that I worked with in the lab. So absolutely, yes, that is so core to what we're building here in Pluto, and have a couple of different ways to do that based on whether you want to interact with an AI or a real person.
All right, great. Well, we are coming up on time here. I want to be respectful of everyone's day. So I think just to wrap up quickly, thank you again, everyone, for joining. As mentioned earlier, we'll make sure that everyone receives a recording of the call that we went through, that you can go back to review, maybe share with colleagues, collaborators, coworkers that weren't able to make it. And as mentioned, we'll share some additional resources as well. There were some questions we were not able to get to just from the sake of time. We'll make sure that we're following up individually on those as well. If you're anything like me, I imagine you'll leave here, 5, 10 questions will pop into your head immediately. You'll see this slide has both my contact info, Ronnie's contact info. We are happy to answer those as they come up, so definitely feel free to reach out. We'd love to hear from you. Also happy to talk through, how Pluto can support some of your research endeavors. Ronnie, I'll hand it back to you to close us out.
Sure. Thank you all so, so much. This is something that we care a lot about, and the feedback from scientists like you is also incredibly valuable for us. So please do reach out, say hi, let us know which parts of this presentation you took away some value from and will be using in your own figure creation. Feel free to reach out directly if we can support you in any way through Pluto, or really just by sharing this knowledge that we've gained from working with so many scientists. Thank you all so much for your time. I hope you have a great rest of your week, and are really coming away, inspired to customize your own figures, tackle those maybe tricky methods sections hunting down that you've been putting off. But go get it. You're all doing such incredibly important work in the world for translating discoveries, and really making those available to the scientific community. So thank you so much to you all, and we hope you have a great rest of your week.
Thanks all.